Good day, everyone. I'm delighted to present a recent publication from our team in iMeta. Our study is titled "With Primer Selection Impacts the Evaluation of Microecological Patterns in Environmental Microbiomes." In the current field of ecology, comprehensively exploring microbial communities in multiple habitats within ecosystems is pivotal for revealing and preserving the biodiversity and stability. Among these, community assembly process has become a hotspot. By employing marker gene primer pairs, high-throughput amplicon sequencing facilitates large-scale monitoring of microbial communities and elucidation of ecological processes. However, in 2021 alone, over 60 primer pairs targeting bacterial 16 sRNA genes were employed. For example, V4 and V5, V7 are commonly used in microbial ecological research across various habitats. Using different primers can lead to different conclusions across studies, and it will also impact our data integration for meta-analyses, which is commonly based on close reference clustering. Previous studies on primer bias mainly focused on specific habitats or species abundance. The primer bias and microecological patterns across different habitats remains unclear. Mulberry Dyke and Fish Pond system provides a stable and controlled model for uncovering primer bias. This study is based on 5 habitat types in this system: water, sediment, soil, mulberry leaves, and silkworm gut, to explore the primer bias on community profiling and microecological patterns. We found that V4 and V5, V7 generated significantly different taxonomic abundance, as well as predicted functions due to their difference in taxa specificity. In addition, positive correlations of primer bias across habitats indicate consistent primer bias of taxa abundance across habitats, suggesting that the primer bias in community profiling can be partially calibrated. At the community level, the predicted functional composition and taxonomic composition were significantly influenced by primers. Although different primer datasets exhibit consistent sample distributions, there exists primer bias in community variation quantification and beta diversity pattern, implying that the choice of primer not only affects the community structure, but also introduces bias in the microecological patterns. In microecological patterns, different primer dataset generated distinct species abundance distribution. In silico simulations reveal that this difference may be attributed to variations in taxa specificity and amplification efficiency. We also found distinct results of community assembly analyses, which might arise from their differential community structure and resolutions, such as longer length and higher entropy of V5, V7 region. Community structure and null model expectation can be both influenced by primer bias, which explains the inconsistent primer biases between habitat types. Furthermore, we found that close reference clustering did not significantly reduce primer bias. This underscores the importance of caution when integrating datasets from different primers and suggests utilizing mitigation strategies, such as abundance calibration, information scale correction, and sample pairing. In summary, our study underscores the profound effect of primer selection on community profiling and microecological patterns. Primer biases and microecological patterns are different across habitats. Thirdly, close reference clustering failed to alleviate primer bias.